Hello everyone, welcome to my channel. And in today's video I'm going to discuss about fragility test. So I'll discuss about the fragility test, which is done to check the fragility of the red blood cells. So it is basically the resistance of the RBCs in hypotonic saline solution. Resistance to hemolysis. Hemolysis means breakdown of RBCs, and fragile means to break. Fragile means to break, so the fragility test is done to check the fragility of RBCs. So sodium chloride solution is used at various concentrations, which is known as hypotonic sodium chloride solution, and the range is 1.2 to 0.2%. First of all, different concentration of hypotonic solution in the range given range of sodium chloride is taken in Con's tubes in a series. To it, 1 drop of blood is added in each tube. After that, it is mixed with the solution. The blood drop of the blood is mixed with the solution and left undisturbed for some time. Now the tubes are observed either directly or by centrifugation process after 15 minutes of duration. So we'll check here the fragility to hypotonic solution, which is known as osmotic fragility, and mechanical fragility is the breakdown of RBCs due to mechanical stress or injury. So the observations are here. In direct observation, if there is no hemolysis, the solution of the test tube appears turbid. If hemolysis started, the fluid is turbid, and if hemolysis started, the turbidity will reduce. And if the hemolysis is completed, the fluid becomes clear. It is the direct observation. And after centrifugation, so we will centrifuge it after 15 minutes duration. So after that, if no hemolysis occurs, the cells will sediment at the bottom, the red cells, and with clear colorless supernatant above. And if hemolysis started, less sedimentation of the cells, and there is a slightly red color of the fluid as the release of little amount of hemoglobin from few hemolyzed red blood cells. And in case of complete hemolysis, there is no sedimentation in the test tube and more reddish fluid as the release of more amount of hemoglobin from all the hemolyzed RBCs. So there is a fragility index for that. So we'll check it after 20 minutes of duration. If there is no hemolysis, it is, and no hemolysis occurs up to 0.6% hypotonic solution, and onset of hemolysis occurs around 0.45% hypotonic solution, and complete hemolysis occurs around 0.35% hypotonic solution. In case of 0.45% hypotonic solution, it is found that the destruction of old cells occurs due to the fragility of the cell membrane of these cells, and younger RBCs are not affected. But in 0.35% hypotonic solutions, the younger cells are also destroyed. So it is a measure to check the fragility of red blood cells, and it is very important to find out certain conditions like hemolytic anemia, where RBCs are destroyed prematurely, or too much destruction of RBCs occurs, which causes the anemia. So here I am ending my today's video. See you in my next video, and if you guys like my videos, please subscribe the channel. Thanks for watching.